So we're going to finish lesson 5.6 and talk about translation. But just to review, we were talking about the codon wheel, the genetic code of life. It's your orange paper that you were given. And we were actually looking at mRNA and how it codes for specific amino acids. So again, all of these, they're on the outside are amino acids. And then starting with the inside, middle, and the last ring, those are your 3 letters of your mRNA molecule. So translation is the actual decoding of mRNA message into this protein. So this is what you don't necessarily see because we were using the codon wheel here on this page, but we have to look and see, like, how does this actually happen? So this is a process that's taking place on the ribosome. So this gray structure here is the ribosome. And you can see the mRNA is going through the ribosome, sort of like you're feeding a paper through the ribosome. You're feeding it through, and as it's going through, there is a transfer RNA. That's what these things are, a transfer RNA that's bringing a specific amino acid to match the mRNA. And attached to that transfer RNA is that amino acid. They're going to string together and form a protein, which is also known as a polypeptide. So if we take a look at this page, let's pretend this whole circle here is the ribosome. We're going to do, again, this is the second part of protein synthesis, the translation part. And this is our mRNA strand, AUG, GUA. The tRNA is carrying what is called an anticodon. It's the other 3 letters that match up. That's how it knows to kind of land in this spot and bring the correct amino acid. Now, I want to point out in blue here, you see that it says you use the mRNA to find the correct amino acid, and you're going to see it matches up right there. Okay, so GUA matches with valine in your amino acid, your codon wheel. So this is going to continue on, and as these amino acids are dropped off, they're joining together by forming something called a peptide bond until they make the entire polypeptide. This is a review of the entire process. So this is your central dogma of molecular biology. Inside the nucleus, you have the DNA. mRNA is made in a process called transcription. mRNA leaves the nucleus. This is a nuclear pore. It goes into the cytoplasm, seeking out the ribosome where the protein is going to be made in a process called translation. Basically, the mRNA is read, amino acids are joined together to form the growing polypeptide or protein. You should practice and be able to draw and explain what is happening and where it's happening in this diagram. And we're going to stop there for today.